Thank you, Phil. First of all, it's really great being here because, as you already said, I did my undergrad here, so it kind of feels like home. Townsville is a little bit of home. And this is now my second year that I came here, and I find it really great to be in the center where you have people working on all kinds of aspects of reef and not just fish and corals and the social aspects. So it's really interesting to be able to come here once in a while. And yeah, so today I'm going to talk about this project that for which we have the first results now. And I'm very excited about this. It's about the response of fish basically to ocean acidification. And I have to start, although I know you're all very familiar with this topic probably, to give you a bit of a reminder of ocean acidification. So a lot of the CO2, the carbon dioxide that we produce by burning fossil fuels is actually taken up by the oceans, and about 25 to 50%. And by doing this, the CO2 reacts with the water molecule, and then the chemistry of the water is changed so that the pH decreases in the oceans due to the production of more carbonic acid. And there are future projections for this ocean acidification. So basically over the next century, all the way to 2100, oh, sorry, that's not the pointer. You see that the pH of the ocean will be decreasing, although obviously this depends on the scenario of how much CO2 we're going to be releasing into the atmosphere. But basically, more or less, by the end of the century, we will have a pH of 7.8 in the oceans, although this varies if you look at the Arctic or, for example, in the tropical regions. And I do want to give you one more statistic here. So for today, nowadays, we have a pH of about 8, and this equals more or less 400 microatmospheres of CO2. And by the end of the century, it's predicted that we will have 750 microatmospheres of CO2. And I'm giving you these statistics because that's basically what we've been doing our experiments in. So I will be talking about the control condition, which is the what we see currently, so 400 microatmospheres, and what I call high CO2 or CO2 treatment. We are talking about treatment of CO2 that will happen in the very near future. But mainly what we're interested in is how does this elevated level of CO2 in the water affect marine animals. And you probably know some of the stories. There are pretty hard effects or crucial effects on calcifying organisms such as corals. And for example, here's just one example of a species that an invertebrate that has a shell, and with more acidic ocean or more acidic water over a really short period of time, the shell can just basically degrade and dissolve in lower levels of in pH. And clearly this has effects on the growth, the survival, the abundance of these species. In terms of fish, we also find some of these effects. So there have been studies showing negative effects of higher CO2 on growth and survival. But a lot of these studies have been done on really high levels of CO2, so not what will be happened by the end of the century, but a lot more CO2. And they have been looking at the immediate reaction to the effect of CO2 exposure. However, when you go ahead and you look at some transgenerational effects, so here what you can see is that so we're talking about offspring length at this stage here, and what happened is that the parents were put into low CO2, so normal current conditions, or into moderate or high CO2 levels. And then they reared offsprings that then were put into low or high or the other kinds of CO2 conditions. So what you see here with the blue bar is that this is how it would be currently. So in a current state of CO2, you would have about a growth of 9.3 millimeters on average for an offspring. However, when your parents are in low CO2 conditions, the current ones, but you as an offspring are born in high CO2, then your length or your growth dramatically decreases. But there's some good news. So if you already have parents that have been living in moderate or high CO2 levels, then when you are born and grow up in moderate and high CO2 levels as an offspring, the length of your growth rate basically goes back to almost control conditions. So this is kind of good news, as it kind of shows that you can acclimate across just one generation with these. In this case, it was offspring growth. But there's also another effect of high CO2 on fish, and it's not just apart from these physiological conditions, it's fish behavior. So fish behavior is impaired, or let's say changed, with high levels of CO2 in the water. And this is done by an impairment to recognize chemical alarm cues. Just a tiny bit on chemical alarm cues. It's something that fish use to recognize distress or a predator, certain things that are going on around them from their conspecifics. So in this case, the Carl Fish, about 50 years ago, 60 years ago, he was the guy who also coined this term Schreckstoff. He found out that one of his fish got injured in the tank, and he realized that all the other fish started reacting in a very different way, and realized that there must have been something that this hurt fish put out into the water for the others to realize that there is danger. Just a quick side note, as this chemical alarm cues have been in discussion in evolutionary biology for quite a long time. Actually, only in 2012 was one of the times where they found or they could isolate one of the compounds that is a chemical alarm cue. And even nowadays, we know that it has to do with olfaction. However, it has to do with some neurons in the brain for which we still don't quite know the function. So we know that that basically they release these chemical alarm cues when they're in distress, and the others receive this alarm by smelling it, but we're not sure how it's connected to the actual brain and the reaction. So on the reef, you've all been there, and you see that there's lots of fish swimming around, and either you grab one, or the predator comes up, or one gets hurt, and then you see that they all retract back into the reef and hide within milliseconds or a few seconds. So that is the reaction of one of them releasing, or several of them releasing chemical alarm cues into the water. So basically, it's very important for them for survival, because if you can't smell it, then you'll be swimming happily around and getting eaten by predators. How do we test this in the lab? Basically, how do we know if they are reacting to chemical alarm cues or not? So you put them into these fluming chamber, or flumes, basically, and you let the fish swim in here in the middle, and it's divided into, or halfway divided into, two sides, and you let water flow in here. That will be control water, just normal water with no treatment in, and on the other hand you would have chemical alarm cue, which would be released by an injured conspecific, and you let that run into the flume as well, which in real life more or less looks like this. This is an anemone fish that gets to choose now if it's going left or right, if it's moving away from the chemical alarm cue, or if it's moving towards the chemical alarm cue. And a normal reaction, you would see that this fish would be moving towards or away from the chemical alarm cue pretty soon. So the question was, we now know that, for example, with growth, there are reversible effects. So if your parents are already in high CO2, you're better off, basically. So do we see these same effects with behavior? And so here, let me go through this graph with you. On the left side, you see the time spent in the queue. So the less you spend in the queue, the more you're able to move away from the chemical alarm cue. And what you see here in the bottom is the parental treatment. So you have control, mid CO2, and high CO2. And the blue ones would be the control offspring. So you see your parents are in control, you as an offspring are in control. You only spend about 10% of time in the chemical alarm cue. So you move away from it, or are in the control condition most of the time. However, when you then move to being here the green ones, as an offspring, you've grown up in mid-high CO2, then you can see that it moves all the way to about 60%, and in high CO2 almost to 80% in chemical alarm cues. So you're very impaired for recognizing this alarm. And unfortunately, you can see that even if your parents are in mid CO2 or high CO2, these bars basically stay the same. So there's no effect of your parents already having been exposed to this CO2. So basically the behavior is impaired no matter if your parents were in CO2 or not. So that means that it's not really good news. However, there is some good news, because when you take individuals from the reef, just from a population, and you bring them back in, and then you test them. So you have some adults, and then you test them, you expose them to CO2, and then you test them in the flume and see if they avoid CAC or if they don't avoid CAC. You will get a variation in behavioral response. So here, it's a bit of a weird percentages, but you have to see here that this is the time spent in CAC, and remember, the less you spend in there, basically the better it is, the more you recognize the danger of it. And so here you have quite a few in the middle, that between 12% and 80% spend their time in CAC. But there are also quite a few individuals that spend very little time in CAC, so 0 to 10%. These are the ones that we term tolerant. So for the rest of the talk, I'll be calling the individuals here tolerant. And then the other side, we have the ones that really are completely impaired and they cannot recognize what the CAC and spend a really long time in the chemical alarm cue. And these are the ones that we call sensitive or non-tolerant, but mostly sensitive. So what we see now is that there is no transgenerational effect if your parents are in higher CO2 or not, but we see that there's a variation in the behavioral response. So with this information, we wanted to basically answer two questions. Our first question was, what are the molecular responses to high CO2 exposure? Because a lot of these things have been studied in sea urchins, for example, but there hasn't been too much done in fish. So we wanted to have an idea of what are the molecular bases that that are involved in this response. And our second question has to do with the variation in the behavioral reaction, and we wanted to see if this variation could also be basically seen in the molecular response of offspring of these two different groups, of the tolerant and of the sensitive group. And this could potentially give us an idea of heritability of this, the tolerance, and then on the other hand, if there is such a signal of inheritance, you could say, I guess, we want to know what is the difference. So what makes this one offspring different to the other. And we did this by doing a transgenerational rearing experiment. So this is now Megan. She took the adult fish that were tolerant, so the ones that spent very little time in CAC, and paired them together in female and male. All of them are more or less the same size, more or less the same sensitivity to CO2. Paired them together, did the same for the sensitive ones, and then either they were placed into control and then gave birth in control, or they were put into CO2 and then were bred also in CO2. So here we have, I'd like to mention a few different groups, a few different colors, because for this group here, which are the offspring of the tolerant parents in control condition, we have 3 family lines. So 3 different pairs that were used for this. And everything I'll be talking about, we're using 3 individuals per family line. So altogether we would have 9 in each of the bigger treatment groups. Okay, so for just a couple slides, you have to bear with me with a little bit about the methods and the molecular analysis that we've been doing. We have been using several levels of molecular analyses, and we go all the way basically from the DNA to looking at the levels of mRNA, which is basically equal to gene expression, and also looking at the translation into proteins and looking at the expression of proteins at for the different conditions. So, like I said before, we've been using the genome. We had to de novo assemble the genome because it doesn't exist. You do need this, and we do need a really decent reference to do this, and that's why I mentioned a little bit here of the metrics, because if you have a bad reference, well, then the results will be a little less of less quality. But we have a really, really good, well-assembled genome. And just to mention a few things, so we have about 25,000 genes, of which 22,000. Oh, okay, sorry, that kind of jumped. Number of genes that have annotation are 22,000. With annotation, what I mean is that we know the function of these genes, and that is a very good statistic, basically, because that means that they are well assembled. Then we sequence 36 brain transcriptomes. First of all, I want to say that we're working with the brain here because we're talking about behavioral responses, and most of them are dealt with in the brain. So the tissue that we use for this is the brain. And we sequence 36 transcriptomes, which is this part of this diagram is the mRNA levels, so it's the gene expression levels. And the transcriptome basically is the total of gene expression across the whole genome. So all the genes that are expressed at that moment, we will see if they are expressed and at what level they are expressed. And we also have proteomes of all treatment groups. Proteomes is this level over here is the level of proteins, and we will know the relative abundance of proteins in each of the treatment groups. Just very quickly to get an idea of how we do these things, we sequence the mRNA, we map it against the genome, and this looks like this. This is the genome, or a tiny little part of the genome. Then I take the sequences that I received from my RNA sequencing, and then I can count and say, okay, look, this is biological sample number 1, it has 5 reads, and then I go on to biological sample number 2, and it has 200 reads. So I know that one has a larger gene expression than the other one. And, this gene expression, basically the more the gene is expressed, the more it is used in this sense. So just to give you an example, if you would have a gene AVT highly overexpressed, maybe in the end you get a lot of vasotoxin, which is a protein that in the end leads to some aggressive behavior. So basically, at higher levels, we find the gene to be more used, and this is similar with the proteome. When we look at the proteins that are all expressed across the genome, we do this a little different. There's no mapping, but we break the proteins up into peptides, we label them, and then by the height of the peak, we can compare the different heights with each other, and we know that, for example, biological sample X 113 has less protein expression than 114. Okay, that is enough on the on the molecular analysis side, but I wanted to explain this just so you know that most of what we're doing is based on the comparison of the expressions of genes and proteins between the different treatment groups that we're looking at. And to remind you again that we were trying to answer 2 main questions. And the first question was looking at what is the difference between the control and the CO2. So basically looking at what is the molecular signature of being exposed or response of being exposed to CO2. And our second question will be looking at the differences between the offspring of the tolerant and the offspring of the sensitive individuals. And to jump into the results, this is a Venn diagram of looking at differential expression of, in this case, of the transcripts, which are the genes. And here I just want to point out to you how that, so for example, here you see, I think it's still blue, right? This blue circle here will be the comparison of control condition with CO2 condition for the offspring of tolerant individuals. And this red one would be the same, so control versus CO2, for offspring of sensitive individuals. And these are the ones I want you to focus on, because we're here looking at the comparison or the response to CO2. And what you can see here, that over here we have 173 genes, over here we have 62 genes, only 7 of them actually overlap in the response to CO2 exposure. And I just wanted to mention a little bit in general about what happens, what is this general response to CO2, and it basically happens at the, in several genes and proteins that are involved in the glucose metabolism, and then we have a few that are also connected with each other that have to do with serine and glycine. So the basic general response to being exposed to CO2 is elevating these levels of metabolism. So it doesn't matter if you are from tolerant parents, it doesn't matter if you're sensitive parents, that's definitely a reaction you're going to have to CO2 in the brain. And now I have to mention something else that was discovered a few years ago from Phil and other collaborators. So they found that when fish behavior is impaired with not impairment, but the interference with a neurotransmitter called the GABA-A receptor. So the GABA-A receptor is one of the main neurotransmitters in the brain. I think it makes up something like 80% of the neurotransmission. And it is an inhibitory neurotransmitter. So basically it stops the transmission of the signal. And this happens with a current of chloride and bicarbonate that then moves through the receptor. However, when the fish is exposed to higher levels of CO2 or more acid in the water, or more acidic water, then this current is reversed, basically interfering with the inhibitory neurotransmission, which means releasing the neurotransmission. And they've seen that, or they have first proposed that this was the cause, and then they actually showed that this was the case, and they did it by using an GABA antagonist called gabazine. So as you can see here, it didn't matter if you put them into control. So this is something similar to the chemical alarm cue. This is the percentage spent in the predator cue, and the control levels were the same. It didn't matter if at control condition plus gabazine, nothing changed. You see that at high CO2, very clearly, you spend a lot of time in the predator cue, which is the impaired behavior. And then then they did this test where at first they just had CO2, then they added gabazine after to the antagonist of GABA, and they saw that actually the behavior went down quite a fair bit, back to about 10%, showing that there definitely was an involvement and interference with this GABA receptor at high CO2 levels. And this was the reason why the behavior was impaired. So in our data here, we can go ahead and look at all kinds of different genes that are involved, genes and proteins that are involved in the GABA receptor or GABA itself. And we see this is just one of the pictures of the expression levels of some transcripts. And you can see that here these triangles is one gene, and the boxes is another gene. So all of them, they're more or less expressed at the same level in the different individuals. So these are all the individuals. So you see more or less it's kind of the same. So there's no differential expression from one treatment to another, which isn't contradictory to the theory that was, or not theory, to the fact that GABA was impaired, because we're talking about a reversal of the current through the receptor, not necessarily an upregulation of the actual receptor or GABA itself. However, we did find on the protein level one protein highly expressed in the high CO2 level for offspring of tolerant parents, showing that there might be, and this protein is involved in the synthesis of GABA, and therefore we could see that there might be a bit, or maybe this might explain a tiny part of why of the tolerance of the parents of these offspring. And what I want to notice here, what I want to show you here is that, so very apparently there is a difference in the reaction to CO2 of of offspring of tolerant parents and offspring of sensitive parents, because there are some things such as GABA that only happen for the tolerant ones. And this can be clearly seen, for example, in this PCA, that we have over here the control conditions and the offspring of sensitive, offspring of tolerant. And when you go over to the high CO2 exposure, you see that the transcription of mRNA or proteins is very extremely different between offspring of tolerant and offspring of sensitive parents. This can also be seen in a way that we usually typically show gene expression, where you have darker colors are higher expression levels. And you can see, for example, here we have the tolerant ones, and here we have the offspring of the sensitive ones at high CO2 level, and you have a whole lot of genes, for example, here, these are very highly expressed for the offspring intolerant, or these ones are less expressed, as in the group that have sensitive parents. So the question is, we see a clear signal, a very different signal, which we also see here in the numbers. When we compare, so here we compare in this, it was blue on here, so you compare the expression in control condition of offspring of tolerant parents and offspring of sensitive parents. I don't know if this is working. Yeah. And then you could do the same thing on the CO2 level, and you see that on the CO2 level you have 152 genes differentially expressed, where at the control level there are very few genes differentially expressed. So the question is now, what are these genes, or what are the different sets of genes involved in the different in the offsprings of the different parental phenotypes? So here we have, I'm going to start telling you a bit about how the offspring of the sensitive parents react. And one interesting thing that came up was that at the high CO2 level, they overexpress histone H1, which is one of the main histones that makes up the chromatin structure. So basically what it does, it takes the DNA and it packs it together. So the more tightly it's packed, the less the less gene expression will happen, because it's basically packed away. You can't get there. So a higher expression in this protein usually shows for a more compact structure in the DNA. However, there's also on the transcript and the protein level, we find some enriched functional categories. And the main one was tRNA amino acylation for protein translation, and these genes such as Rs, DARs, CARs, they're all involved in a really important, actually extremely important, for the translation from mRNA to proteins, as they pick up the correct amino acids and give them to the tRNA to translate into proteins. So the fact that they are overexpressed in this group, the group of offspring from sensitive parents at a high CO2 level, well, could show that they're trying to, let's say, make up for something that they're struggling with. However, on the other hand, this could become maladaptive, though. And then I would like to mention something else, because these tRNA synthetases aren't only differentially expressed, but we also found a difference in the genomic sequence of these genes, or of some of them at least. And just to give you a quick idea of what I'm talking about when I say differences in the genomic sequence. So here, for example, this is just a chunk out of a sequence of one of the offsprings of the tolerant parents, and this is a chunk out of the sequence of an offspring of the sensitive parents. And you see here you have, on one side you have a T, and on the other side you have an A. And by looking at, doing Bayesian analysis, you could see that if many of these tolerant individuals have a T here, and many of these offspring of sensitive individuals have an A here, there's a difference in this, and you could call it, well, an evidence of selection. And we were looking for these differences in the genomic sequence that we call SNPs, single nucleotide polymorphisms, which is just one particular spot in the genome. And we found that 4 of them were so-called outlier SNPs that had some evidence of selection, and 2 of them were in some immune-related genes, and the other 2 were related with the, as I just mentioned before, the tRNAs, which is the ones that help the translation into proteins. And so both of them had a different SNP, or, well, was a different, basically had a different base in that position. And this could mean that for the sensitive, or the offspring of the sensitive individuals, these genes might become maladaptive. And then switching over to probably the coolest part, I think, is, so what defines the tolerance? Now we know, okay, there's some certain things that the individuals, the offspring of sensitive individuals do, but what happens to the ones that come from tolerant parents? Well, in this case, we find all the genes and a lot of proteins upregulated or differentially expressed for the circadian rhythm. And I just, before mentioning anything else, I wanted to let you know that usually altered levels of the circadian rhythm, especially if they're differentially expressed from one treatment to the next, show that there is a phase shift in the circadian clock. I'm assuming that everyone has heard, of the circadian clock, right? So there's a 24-hour cycle, more or less. It's not quite 24 hours, but so a lot of our processes, human, any other animal, they follow these cycles quite closely. And so in this case, when you have altered levels of the circadian rhythm genes, you can invoke a phase shift in the circadian clock. And this often allows for an adaptive advantage. If you're faced with some environmental change, for example, let's say usually if the days get shorter, a plant will also start changing their circadian rhythm just to be able to use the light more, for example. So there's environmental change, you can phase shift your circadian clock. And this is just a very overwhelming diagram, I think, of the circadian rhythm. What I want you to see here is that there are many, many genes involved, and there are activator genes and there are repressor genes. These ones here in the middle are the main activator genes, and you have a lot of repressor genes, which are the ones in red. And the takeaway message of this complex diagram is that all the red ones, which are the repressor ones, have this little green star, which means that it is overexpressed for the offspring of tolerant individuals at high CO2. So basically, for the offspring of tolerant parents at high CO2, the circadian rhythm is downregulated, meaning that it allows for a phase shift in the circadian clock. So what does that help those individuals? Well, as you might know, fish have acid-base mechanisms, and they really need to regulate their acid or have acid-base balance. And in the face of high CO2 or ocean acidification, one of the main things that have been shown that are really important is that individuals have to avoid acidosis. So they have to avoid the fact that there's so much more acidic water, or in general, there's more acid coming into the system. So one of the things that they have to avoid, and most of the fish closely regulate their ions that are relevant to the acid-base control. And they do this with a response to environmental fluctuations in CO2 in correspondence with the circadian clock. So in the case of offspring of sensitive parents, we just see that they normally regulate their circadian clock. There's nothing different to it. Plus, they have a lot of other genes, which I didn't mention, involved that, like melatonin, you might have heard, something you might take if you have a jet lag. You know, it's a control mechanism also of the circadian rhythm. So these guys display a more pronounced iron regulatory adjustments, which basically makes them really good at regulating their ions. So they're good at it, but on the other hand, this leads to a more altered current through the GABA receptor, as they're really tightly regulating their acid-base control, and therefore, well, then you end up having an impairment of their behavior. And on the other hand, we have the tolerant parents, or the offspring of the tolerant parents, that show this downregulation and phase shift in the circadian clock, allowing them to basically be a little bit more relaxed about controlling their acid base, and therefore not so and therefore the flow through the neurotransmitter is not it's not so profoundly altered, let's say. And so basically, the ability of this flexibility of controlling the acid-base control through the circadian rhythm probably makes these offspring of the tolerant parents less susceptible to behavioral impairments. And just to sum up a few things that we've learned in our study, well, we have a whole bunch of genes and proteins that are differentially expressed from control to high CO2, as well as between the two groups coming from the different parental phenotypes. The general response to high CO2 is the involvement of genes and proteins associated with the glucose, serine, and glycine metabolism. And really interestingly, well, the majority of the difference between the offsprings of tolerant and sensitive parents occurred at high CO2, not a control. It only occurs at high CO2. And consequently, we see a signature of the parental sensitivity to the high CO2 in the molecular phenotype of the offspring, which is primarily driven by the circadian rhythm genes. So I just want to make a little bit of a point on the importance of the possible inheritance, and that I think we might not have to put gas masks on all our fish now, because the fact that we see such a signature, although this needs a lot more study and something that we'll be doing now soon, the fact that we see the signature in the variation that seems to be, well, not inherited, but we see a signature suggests the possible inheritance of the CO2 sensitivity, and this could help populations to adapt to ocean acidification, basically due to a certain variety or certain variation being able to deal with high CO2. So what we're trying to do now soon is we would like to get to the bottom of what is this variation. So we know that there are a whole bunch of genes that are, especially circadian rhythm, etc., that are really important with dealing with high CO2. But we would also like to understand if it's, and there are some SNPs that we saw, but we would like to see if it's a genetic variation or how large the epigenetic component here is. And we will be looking first at the differences in genetic variation between the parents this time and the offspring of tolerant and sensitive individuals, and we'll be doing this by genome resequencing, so we'll be going down to the base level and have whole genomes for each of the individuals, and then try to find out what parts of the genomes are different and what parts or what leads to this difference in variation. And then also the other interesting thing is that we don't only have offspring of parental pairs that were tolerant and sensitive. We also have one male was sensitive and the female was tolerant, and the other way around. So we can also look at is it more driven by mothers or fathers, or basically females and males. So I would like to thank a whole bunch of people. First of all, of course, Phil and his group here, Meg, who did all the experiments, Heather, who's my to-go person for absolutely everything, And then my group, the people I work with in Kels, it's Tim, and then Tivu, who has been involved in the genome assembly. Also Göran, who is in Norway, who helped us a lot with interpretation of which protein is important and which gene, and understanding the GABA receptor. And I'd like to end up with let's hope that fish don't have to, do this in the future, and they're going to survive.